Thank you very much for inviting me to speak. I really appreciate it. So I'm going to tell you where we are with CARD14, 15 years on. So I'm going to tell you about how we identified this gene, which is mutated in rare forms of psoriasis, and which we believe actually plays an important role in common psoriasis. Tell you about the different inflammatory skin diseases with mutant CARD and sort of compare and contrast them. Tell you about the signaling pathway that CARD14 activates in psoriatic skin, not surprisingly, NF-kappa B and MAP kinase, and mTOR, which I'll tell you about. Some recent work we've done with Steve Lay on looking for the components of the CARD14 signalsome, which have also told us a bit about what it does. And then some recent work in my lab where we're trying a new form of psoriasis treatment, which is to degrade CARD14 with PROTAC. So this whole story started many years ago. It was the brainchild of Alan Mentor, who had been advised by people like Arlene Auerbach, people working with the MPF, that the way to understand psoriasis was to understand the genetic basis of the disease. And so we started a tissue bank, a psoriasis tissue bank, when I was in Texas. There's Dallas. And this is from 1994. The National Psoriasis Tissue Bank opens its door. And this here, this photo is actually a little girl. She's 12 there. She had psoriasis and psoriatic arthritis and was part of this large family that turned out to have CARD14 mutations. So we went and collected these large families. We looked for what was then linkage analysis, so mapping the genes. And most of the families were too small to conclusively map a single gene, but this one family came across my desk, and it's a family from Kentucky in C. This pointer doesn't work, by the way. No. Okay. So the C family is, oh, I'm sorry, B. So B is a family from Kentucky. All the affecteds are in black. There's over 20 affecteds. It looked like an autosomal dominant trait. And we also, once we thought we'd found the gene, which took a very long time, 17 years, because the genome project wasn't achieved at the time, we went back and looked at a Taiwanese family where there had been mapping of a psoriasis locus to chromosome 17q. So this is separate from the MHC, and in fact, you don't need the MHC in this case, HLA-CW6, to get disease. And we also showed that the mutations in both of our families were at the same site. So they actually introduce a novel peptide into the protein, which we now understand why this leads to inflammation in the skin. And this was done by Catherine Jordan, who was an MD PhD student in my lab, along with other people that were there at the time. So we also were working with Jim Kruger and Michelle Lowe's to stain for CARD14 in the skin. And Michelle said that she knew of a child from NIH being looked after by Rafaela Goldblatt-Mansky, who had severe generalized pustular psoriasis. And I don't know why, but Michelle thought they may have a CARD14 mutation, and they actually did. And the mutation was a de novo, so it wasn't in either parent. Here were the sequencing traces, and you can see the proband. And that led to a glutamic acid to alanine change at position 138. Now, this mutation has been incredibly useful for many downstream activities. We've made mice with that mutation where we can recapitulate the skin disease. And we've also now used keratinocytes of this child to start assaying for novel small molecules that will modulate the pathway. So we did a lot of resequencing of this gene in collaboration with many people, many groups, psoriasis groups, including JT's group here, many others, Wilson Liao, and we found mutations throughout the gene, but in psoriasis cases, but they clustered in the coil domain of the gene. So we also found that there was a common change, so an allele that is present at 50% about in the population that when you had the one amino acid, it increased psoriasis risk, which puts CARD14 not just in these sort of the pathways leading to rare forms of psoriasis, but actually to common disease as well. And how these interact with other common variants identified through genome-wide association studies isn't clear yet, but hopefully eventually that will get worked out. So there was also a study done by Koto's group showing that both common and rare CARD14 variants affect the anti-TNF response among patients with psoriasis. So it started to have some clinical implications. So CARD14, so where was it expressed? So it turned out that it has very limited patterns of expression. It's expressed in epidermis, in the keratinocytes. Here you see uninvolved skin, involved skin, where it actually is expressed throughout the suprabasal and granular layers, whereas in non-lesional it seems to be expressed at the more basal or maybe suprabasal levels. We also found that if you go to a database called GTEx, you can see where it's expressed. And it's interesting because this and some other genes that we think are mutated in certain forms of psoriasis have very similar patterns of expression. So the blue, these are all different tissues down the bottom on the x-axis, and the blue actually is skin. But then we also have esophagus, where that's the brown, and so mucosal epithelium, and also vagina. So there's something about its pattern of expression, I think, that we will maybe understand more about the selection pressures associated with these mutations based on the patterns of expression in future years. So Jamie Harden, working with Jim Kruger, also showed that CARD is expressed in cells of the dermis, and these are endothelial cells. And we knew that CARD14 plays a role in NF-kappa B activation from earlier studies by Burton and co-workers. And she could show that CARD14 co-stains with endothelial cells and active NF-kappa B. So two different cell types. The other thing is that the family where we mapped the gene originally has not just psoriasis, the affectors can have psoriatic arthritis at about the frequency in the general population, which said to us that those were the risk of psoriatic arthritis in this family was modifier genes rather than other mutations. And we haven't identified those. That's a real challenge. So others started looking at inflammatory skin diseases with CARD14 mutations, and soon after we published our finding of CARD14 in familial psoriasis, Eli Sprechter showed that CARD14 mutations are found in familial pityriasis rubra pilaris, a very similar kind of phenotype. And then later, working with Keith Choate, we were able to show that CARD14 is mutated in certain pediatric psoriasis, so a disease called CAPE, CARD14-associated papulosquamous eruption. And so this had features of both psoriasis and pityriasis rubra pilaris and certain characteristic features on the face and so on. So there have also been other studies. For example, one study showed that whereas CARD14, when active, leads to enhanced NF-kappa B signaling in the keratinocyte, that loss of function mutations in CARD14 are associated with atopic dermatitis. So whether this will pan out in larger data sets it'll be interesting to see. And also, if you look at patients with CARD14 mutations, you can have they can have classic psoriasis, generalized pustular psoriasis, this CAPE, and then also in Asia, there's a form of psoriasis, GPP, with classic psoriasis, and it's associated with a particular amino acid change. So some of the mutations seem to be responsible for the severity of the disease, and with the most severe being generalized pustular psoriasis. So I started working with Steve Lay when he was at UCH, and postdoc Ashley Howes. They then moved to the Crick, and we're trying to work out how CARD14 mutations lead to psoriasis. And so what we've been able to show is that CARD14 on the left folds back on itself in a quiescent state. And when CARD14 is mutated or possibly activated, as in classic psoriasis, it opens up, maybe because of phosphorylation events, and it recruits two proteins, BCL10 and MALT1. And this forms what's called the CBM complex, standing for CARD14, BCL10, MALT1. And then that leads to activation of NF-kappa B in the keratinocyte. And so here we have the constitutively active CARD, where it's always bound to BCL10 and MALT1 in, for example, this child with the generalized pustular psoriasis. Now, one thing I'm very interested in is whether CBM complexes form during classic psoriasis, and I suspect they do, that's part of the whole mechanism going on in the keratinocyte during inflammation. There's also a ton of proteins that come in and sort of downregulate the response, which is why the disease then can wax and wane. So we came up with a hypothesis for psoriasis pathogenesis in these patients with mutant CARD, that mutant or activated CARD forms a CBM complex that also relies on molecules such as ACT1 and TRAF6, that this then leads to NF-kappa B and MAP kinase activation, the production of chemokines such as interleukin-36 gamma, interleukin-8, CCL20, which recruit immune cells, and then some of these, like T cells, differentiate to Th17 cells, and then produce IL-17 and IL-22 that acts back on the keratinocyte, so you get this vicious cycle of disease, of inflammation. And of course, ustekinumab, which was being used at the time and was very efficient in this child with generalized pustular psoriasis, targets one of these, a part of this pathway, because it's targeting the interleukin-23 molecules. And now, these other downstream molecules like IL-17, also targeting them, also work well for CARD14-associated psoriasis. There's also a TNF branch, which this whole pathway activates TNF as well. So we asked, one of the acid tests of whether you found a true mutation is to make it in a mouse. And so we have made several mouse models of CARD14, mutant CARD14. And so the first one we actually made with John Sumberg at Jackson Lab. So we knocked in this E138A mutation. The initial mice that were born they died at birth. So we crossed them to other mice. This one strain now has survived on an FVB background. So it's nice and stable. There were other reports from Mellett, for example. They made a CARD14 delta E138, so they deleted that amino acid. There was a paper recently from Wang, where they made this mutation and a delta Q136. These are all in the heterozygote. So homozygous mice, actually, for them, the disease is much more severe and sometimes lethal. And then we worked with Steve Lay and Juan Manils, and we made a keratinocyte-inducible mouse, CARD14 mouse. And so here you can see, for example, the thickened tail after treatment with tamoxifen on the right. So the classic epidermal thickening of the tail. And here you can see the ears, with the ear on the right being the heterozygote at 1 month. So these mice have the classic, phenotypes: scaling skin lesions, epidermal thickening, keratinocyte hyperproliferation, hyperkeratosis, enhanced activation of the IL-23, IL-17A axis. The immune infiltrate is rich in neutrophils, myeloid cells, T cells. And what's also interesting is that Tanaka and co-workers knocked CARD14 out in mice, and then when they tried to develop lesions with an imiquimod model, they didn't get lesions. So in other words, knocking CARD14 out may be a way of treating disease, and I'll get back to that. So we also worked with Steve and a graduate student of his, Paul O'Sullivan, to ask what the CARD14 signalsome was. So what proteins are binding CARD? So we took the mutant CARD again, the E138A, and did mass spec and looked for interactors. And so on the right, you see at the top some of the interactors that came out. And a few, of course, have come up before in psoriasis genome-wide association studies, like TNF-AIP3, TNIP1. We also pulled out MALT1 and BCL10, so that was good. And then Paul looked for the CARD14 phosphoproteome and asked what proteins are phosphorylated with mutant CARD. And you can see that one of the genes that came up is Raptor. And so this is a component of mTOR signaling. So it's involved in keratinocyte metabolism. And I don't have time to take you through the data, but it looks like CARD14 mutations both activate the immune system and independently change keratinocyte metabolism by activating mTOR. So what we also determined, so a number of these interactors on the left are components of NF-kappa B signaling, and you can see all the proteins that have come out. But what was also very interesting was that we identified components of the early endosome, and those are on the right. So it looked like CARD localizes to early endosomes, and it's associated with the AP2 adapter complex, which is required for activation of mTOR complex 1, stimulating keratinocyte metabolism. And as you'll hear from Francesco in a minute, one of the components of this is AP1S3, and that is mutated in some forms of generalized pustular psoriasis. So a role of the early endosome seems to be important in psoriasis, which is different from the other CARDs, like CARD11 and CARD10, which are very similar to CARD14. They don't seem to interact with this complex. So the other question we asked, given what we knew about CARD14, is I know we don't really need to treat psoriasis right now. There's fantastic drugs. But could we treat psoriasis by degrading CARD14? And so if you have an activated CARD14 protein, it recruits BCL10 and MALT1. That leads to inflammation and proliferation and to psoriasis. But what if we degraded CARD14? Then the whole complex would fall apart, and we wouldn't have inflammation. So we've been doing this. We've been working with a chemist at Mount Sinai called Jian Jin, and he's developed a method. So there's a method called a PROTAC, which stands for proteolysis targeting chimera. And so what it does is you identify small molecules with PROTAC that bind a protein of interest and take it to the proteasome for degradation. But because CARD14 doesn't have any small molecules that interact with it, we're doing what's called bridge PROTAC, which is where we actually now have molecules that bind MALT1, which is part of the CBM complex, so activated CARD. They've identified small molecules that will interact with MALT1 and will also take this complex to the proteasome. So the goal of this whole study has been to identify small molecules that degrade CARD14 over MALT1, because of course you're taking the whole complex to the proteasome for degradation. So I just want to show you some of those results. So what we've used for our assay is the keratinocytes of the child with the very severe generalized pustular psoriasis and the E138A mutation. And so these are just assays for NF-kappa B targets, so CCL20, IL-36 gamma, IL-8, and SOD2. And on the left, you see the results with healthy keratinocytes, and then on the right, the LAF line is the levels of, for example, CCL20 in that line. That's just constitutive, no stimulation, nothing. And so we thought, if we treat with these degraders, maybe one of them will downregulate, or some of them will downregulate this activated, this NF-kappa B activated pathway. So here's the results of the first 29 degraders. So our goal is to look at 150, and we actually have a grant from the NPF to do this, to start doing this. And so here what we're looking at is levels of NF-kappa B activation in untreated cells, and that's the 1 value. Okay, so this is all normalized to untreated cells. And then here's the levels of NF-kappa B activation on the basis of NF-kappa B targets relative to untreated cells. And the samples that are boxed in red are the the small molecules that seem to consistently downregulate NF-kappa B. So there's a lot of work to do. We have, a ton of work, like toxicology assays and, assays in mice and so on, but I'm excited about these molecules, and who knows? So what have I told you? I've told you that rare gain-of-function mutations in CARD14 can lead to classic psoriasis, psoriatic arthritis, generalized pustular psoriasis, K. pityriasis rubra-pilaris, and hypermorphic mutations in CARD14. So mutations that lead to low levels of NF-kappa B activation may be associated with atopic dermatitis. The mutant CARD14 leads to the formation of a CBM complex, which consists of CARD14, MALT1, and BCL10, activation of NF-kappa B and MAP kinase signaling in keratinocytes and endothelial cells. And CARD14, the mutant CARD14 localizes to early endosomes and is associated with the AP2 adapter complex, which is required for activation of the mTOR complex 1 stimulating keratinocyte metabolism. And I've told you about our early results with degrading CARD14 via bridge PROTAC, and those are early results. But since we know that mutant CARD14 can also lead to psoriatic arthritis, maybe it's a novel way of treating psoriatic arthritis too. So just to acknowledge everyone over the years, my group over the years on the left, Jim Kruger, Wilson Liao, JT, Alex, people from Taiwan, Raphael Goldblatt-Mansky, John Sundberg, people at Imperial, and Alan Menter in particular, who really was the brainchild of all this. Thanks.
I think we're running short on time. I think we have time for one question. I have a quick one.
Yeah.
I think Andy's hand went up first.
So has the genotype phenotype been worked out, because the phenotypes are different, right? Psoriasis, pustular psoriasis, PRP. Do we know anything about the genotype associated, let's say, with psoriasis mutant versus PRP patient? Are the genotypes different?
So the PRP is a challenge, but certainly for psoriasis, the most severe cases, if you do NF-kappa B assays, the levels of the NF-kappa B targets are by far the highest. So this child that I showed you with the severe GPP, she produces more IL-8, for example, than any other case. So there's certainly a correlation with levels of NF-kappa B targets and severity. But how you differentiate PRP versus psoriasis, I suspect it's genetic modifiers. I mean, we did have a few cases in the family, the big family from Kentucky with the one mutation, where some of those cases had HLA-CW6, and they also had developed psoriatic arthritis. So the question was this sort of an additive effect?
Okay.
But
and then the CARD14 knockout mice, do they get eczema? Are they prone to get it? Because obviously you'd be worried about degrading CARD14 and causing eczema, right?
Sure.
Do the CARD14 knockout mice?
The knockout, I don't know if they got eczema. But you see, the great thing with the small molecules is you can titrate them, because that's what you want to do.